Hello again. This time I will be talking about macroinvertebrate export from our type N study streams. Macroinvertebrates are a variable of interest given their role as a food source for amphibians and for downstream fish. We hypothesized that timber removal would decrease leaf litter inputs, which would then increase the number and biomass of macroinvertebrate shredder taxa, which feed on leaf material. We also hypothesized that timber removal would increase solar radiation, which would increase periphyton growth, and the numbers and biomass of macroinvertebrate scrapers, which graze on periphyton. We assessed macroinvertebrate export from the 8 study basins. That included the 4 in the Olympic block and the 4 in the Willapa 1 block. We limited sampling to these 8 basins because of the time required to collect and process the samples, and also because these 8 basins were being monitored for flow, and we needed flow quantification to interpret the export results. We sampled macroinvertebrates from the drift in conjunction with detritus sampling. The samples were collected near the flow monitoring equipment about every 6 weeks for a 24-hour period, although we had to sometimes shorten the sampling period because of high flows. The sampling net consisted of a 250 micron mesh net with a drawstring design, and we wrapped the net around the lip of the flume and tightened the drawstring, and then further secured the net with zip ties. In the lab, we rinsed the drift samples through a series of nested sieves and separated the macroinvertebrates from the detritus. We identified most macroinvertebrates to the lowest practical taxonomic level. Some of the macroinvertebrates, such as the chironomid midges, we left at higher taxonomic levels. We measured each individual to the nearest 1 millimeter and then used published taxon-specific length-weight regressions to estimate biomass. We calculated macroinvertebrate export rate by standardizing numbers and biomass to 24-hour sampling day to give numbers per day in milligrams per day. We also used the stream flow measurements recorded by our hydrology instrumentation to calculate export density in numbers and biomass per cubic meter of stream flow. We summed these 4 metrics by total macroinvertebrates, functional feeding group, and dominant taxa. Because export is flow dependent, we calculated a treatment effect between the treatment and its respective reference over the same period of time, and then averaged the treatment effect by treatment period for each of the metrics. We log transformed the data and used a generalized linear mixed effects analysis of variance to analyze the data. We ran post hoc comparisons to see if there was a difference between the treatments. This plot illustrates the result of the post hoc comparisons for total macroinvertebrates exported in numbers per day. Each comparison shows the difference and the direction of change between the pre and post harvest period and between the different treatments. The comparisons with 95% confidence intervals that do not overlap 0 indicate a change in the post period and between the treatments. And we can see here that there was no change in total macroinvertebrate export in numbers per day in the post period for any of the treatments or for between the treatments. We also did not see a change in total macroinvertebrate biomass exported per day and in numbers and biomass exported per cubic meter of stream flow. The mixed effects model did detect a change in macroinvertebrate predators. This post hoc comparison shows the export of predators in numbers per day, and we can see that predators decreased in the 0% treatment in the post period. We also saw the same pattern for predator export in milligrams per day and in numbers in biomass per cubic meter. There was no change between the treatments. The mixed effects model also detected a change in macroinvertebrate collector-gatherers, and this post hoc comparison shows an increase in collector-gatherer export and biomass per day in the 100 FP and 0% treatments in the post period. There was no difference between the treatments. And here's another post hoc comparison showing collector-gatherer export and biomass per cubic meter. And this shows an increase in collector-gatherer export in the FP treatment in the post period. We ran the mixed effects model on all the different functional feeding groups, and it didn't find a change aside from the collector-gatherers and predators, it didn't find a change in any of the other feeding groups, which included collector filterers, scrapers, and shredders. This is the feeding group composition of macroinvertebrates exported in numbers per day. Pre and post, treatment and treatment period are listed on the x-axis, percent composition on the y-axis, and the functional feeding groups are listed here on the right. For those of you unfamiliar with macroinvertebrate functional feeding groups, shredders consume leaves and other larger particulates, scrapers graze on periphyton, collector-gatherers and collector filterers consume the smaller particulates that were processed by the scrapers and the shredders, and the predators eat other macroinvertebrates. Chironomids can be either collector-gatherers or predators, depending on the taxon, but because we didn't identify them past the family level, we couldn't assign them to the feeding group, so they've become their own category. We can see here that the chironomids in blue and the collector-gatherers in green made up most of the proportion of the export in the pre and post period in the reference and in the treatments. Here's functional feeding group composition of the export in biomass per day. Collector-gatherers in green and predators in purple seem to make up the bulk of the biomass for most of the treatments and treatment periods, although we did see contribution from filters here, maybe shredders here, maybe scrapers here. But if you recall, the mixed effects model did not see a change in any of those groups. We also ran the mixed effects model on some of the dominant macroinvertebrate families. Some of these included the chironomids, the simulids, the dixid for the true flies, the baetid and heptageniid mayflies, and the nemurid and peltoperlid stoneflies. And this comparison shows that baetid export and biomass per cubic meter increased in the 100% FP and 0% treatments in the post period. There was no difference between the treatments. This is the only one that came out that we saw that changed after harvest. None of the other dominant taxa changed. This plot shows the taxonomic composition of the export in numbers per day. We have treatment and treatment period on the x-axis, percent composition on the y, and the major macroinvertebrate orders are listed here on the right. Coleoptera are beetles, Diptera, true flies, Ephemeroptera, mayflies, Plecoptera, stoneflies, and Trichoptera, caddisflies. The Diptera made up probably the bulk of the export. Those consisted mostly of the chironomids. Ephemeroptera also made up a lot of the exports, specifically in the post-harvest years, and that consisted mostly of baetid mayflies. Diptera and Ephemeroptera also made up a large proportion here in the reference in the post period. And then we also see this prevalence of Trichoptera in these in the pre-period and the reference in 100% treatment. And this was due to a collection of a large amount of early instar limnophilid caddisflies during a couple of high flow events from the Olympic block sites. This is the taxonomic composition of the export in milligrams per day. This one doesn't really seem to have that much of a pattern, although we can see that Diptera, Ephemeroptera, Plecoptera, and Trichoptera were all well represented, whereas the beetles in blue and dark blue really weren't. So conclusions, we didn't see a change in export rate and density for total macroinvertebrates and for most feeding groups and dominant taxa. However, we did see a decrease in predator export rate and density and the 0% treatment, and an increase in collector-gatherer export and biomass per day in the 100% FP and 0% treatments. We also saw an increase in collector-gatherer export and biomass per cubic meter in the FP treatment, and an increase in bayetid export and biomass per cubic meter in the 100% FP and 0% treatments. Of the major functional feeding groups, chironomids and collector-gatherers made up most of the individuals exported per day and per cubic meter of stream flow. Collector-gatherers and predators made up a large proportion of the biomass exported per day and per cubic meter. Of the major orders, Diptera and Ephemeroptera comprised most of the individuals exported per day and per cubic meter. So if you recall, we hypothesized that timber removal would reduce leaf litter inputs and potentially macroinvertebrate shredders, and timber harvest would increase solar radiation, which could increase periphyton growth and the production of macroinvertebrate scrapers. So why didn't we see that hypothesized community structure, change in community structure? We think that wood may have helped may have contributed to this lack of change in the community structure. The basins received a large amount of wood during harvest, particularly the 0% treatment and the unbuffered portion of the FP treatment in the form of logging slash. And then over the years, we also saw that a lot of the buffer trees blew down in the FP treatment and in the 100% treatment. And this picture here is from the Willapa 1 100% treatment site. And you can see the wind throw in and over the channel, and the channel's flowing right through there. So we think that maybe the wood played a role in creating debris dams and depositional habitats, which may have acted to trap what organic matter remained in the stream system for food for macroinvertebrates. Other studies have shown that the addition of wood to streams enhances detritus retention and also increases the abundance of collector-gatherers, specifically the chironomids and the mayflies. And also, the dams may have, the slow water habitat created by the dams may have caused an accumulation of fine sediment, which, when combined with shading from all this additional wood, may have prevented periphyton growth and the production of macroinvertebrate scrapers. Another thing to note, too, is that two of the families, the chironomids and the bayetids, were well represented in a lot of the samples. And studies have shown that taxa from both of those families are prolific drifters and adapt pretty quickly to disturbances in streams. And that can come from their opportunistic feeding habits, but also the fact that they're multivoltine, and that means that they're able to cycle through multiple generations in a year, which puts them at an advantage over some of the other taxa, like the large predator stoneflies that can take a year or more to cycle through a generation. And that's all I have.